All right. All right. Can everyone see the screen?
Yes. Yep.
Perfect. So again, welcome everybody. Um, for those, if you may have missed the first meeting, I encourage you to go back and watch that recording. Um, oh, Nicole, did you have a question? All right, if you have one, you can just put it in the chat. Um, but yes, welcome everyone. This will be our meeting on reviewing the paper for this semester. So it'll mainly be about understanding what's going on in this paper, the motivations for this paper, um, and also um, forecasting what we will be doing with this paper in the future a little bit as well. So to get started, the paper that we're talking about is a computational model of the escape response latency and the giant fiber system of Drosophila melanogaster. So this is a fruit fly, and we're going to be looking at how does this escape response latency, or how fast the fruit fly signals that it wants to escape, and how that changes as the fruit fly ages. And then not only how it changes, but what specifically about the neurons change um, as well. So before we get too deep into anything, I think it's good to have a big picture of what is going on, because otherwise you might feel lost in the weeds. So the big picture is we're figuring out what causes this increased latency as the fruit flies age. So again, we're looking at this escape response latency, because it might be important for these fruit flies to have a small latency so that they can get away from danger quickly. And then we'll be investigating how the circuit changes, and we'll be looking at different features, so its physiology or anatomy. And then this can highlight potential future experimental targets in other organisms like rats and even humans. So this is what we'll be talking about today. So we'll again go into the motivation of why should we even be taking a computational neuroscience approach to this problem. We'll be giving some background information for some terms that are used in the paper, and then we'll have a chance to brainstorm about potential things that could be changing. And then we will briefly touch on modeling with neurons, but that'll be for a future workshop. And then we'll talk about some of the results, and then we'll wrap up. All right. So why computational neuroscience? So computational neuroscience, we're going to be looking at the biology of the neuron, its physical properties, and then writing some sort of code and using math to represent this circuit. So all of this entire model that we will be investigating is all based off of real physical properties of these fruit flies. Um, and the reason why that we want to do it with computation is that it allows us to investigate properties that you wouldn't normally be able to change in fruit flies, and to also do more replications of it. And also, this greatly reduces the cost of the experiment and the time to do it, um, especially when you're looking at something like aging. So here's a picture of the fruit fly and what it looks like. And you may be asking yourself, like, how is a fruit fly related to humans? Why should we even spend time focusing on a fruit fly? Um, the reason why is because they've been studied extensively, so for more than 100 years. So all of the neurons and the genes of these fruit flies are mapped out, which is helpful for us. And then they also have a short life cycle. So specifically when you're studying aging, you don't want to have a long life cycle because that's just going to prolong your experiment, make it more expensive. So this allows us to study aging better as well. And then they actually have a high genetic homology to humans. So about 75% of the disease-causing genes are conserved in humans. So although we look very different, um, some of the same themes are present across organisms. And compared to other organisms like rats, they're also more inexpensive, easy to maintain, and they also have a smaller number of neurons. So it's easier to map out a whole circuit where you need as much neurons as possible. Um, so down here, we can see a picture of them mapping out these neurons in the fruit fly. And why are we looking at the escape response latency? So why do they choose to measure this? Um, you can imagine that um, if we weren't, if they had too long of a latency, then they wouldn't be able to escape a predator, meaning that they would um, be swatted, like we see in this little GIF here. And all of the latencies, or another word for latency is the time from when the neuron is stimulated to when their muscle twitches, signaling that, okay, they're going to move. That's the time that they're measuring. And that's the time that's increased in um, aging fruit flies, which causes an issue because that causes a slower response. So then they're not able to escape. So that's why we're studying these metrics. Okay, again, how does this relate to humans? So the core question that this experiment is investigating is, why do neural circuits slow down as an animal gets older? You know, that's something that can apply to all organisms, including humans. And then what are the mechanisms behind this decline? So these strategies that are used um, when studying these circuits in fruit flies can also be applied to humans, and then even some of the properties might be targets that we want to investigate in humans. You know, it's better at least researching it in a fruit fly before starting in humans than just going into humans blind. So that's kind of the whole motivation of using this. But we also have to understand that there are limitations and there are differences, um, and to take these findings with a grain of salt and to trust them but verify them first, you know? All right, so now we're going to go into some background information. Before I continue, does anyone have any questions at all about anything? Oh, I see one in the chat. Perfect. All right. Okay. So the circuit that we're studying is the giant fiber system in the fruit fly. So this is the anatomy in the paper. If you would like to, you can follow along with the paper. I posted the link in the chat, the WhatsApp chat, so you can access it there. And this diagram is in the paper. But what we're seeing is that these giant fiber interneurons are descending from the brain and into the thorax. And then this circuitry is going to be involved in two actions, jumping and flying for the fruit fly. But first, let's talk about some more um, specifics about the anatomy. So here is a simplified diagram that they created. And this rod right here represents the giant fiber that we saw earlier. And you can see that this is where they're stimulating. So then we're seeing that latency to when it goes to these other neurons and then to the muscles. So we have the giant fiber here, and that's abbreviated as GF. And then we had the turbotroch and netral muscles, tongue twister. And we have that shown in blue down here. And then we have the DLM, the dorsal longitudinal muscles, depicted here. And then we also have the PSI, which is the peripheral synapsing interneuron. Um, Another thing to take into account with this diagram is that they also have the synapses labeled. So we see it's going to the giant fiber, and then we have an electrical synapse with the PSI, and then also down to the giant fiber, an electrical synapse to the TTM, and then there's a chemical synapse, which is with the DLM. So the general pathway for the jump pathway is we're going to go from GF, TTMN, and the N um, abbreviation just means motor neuron. So TTMN to TTM muscle. And then again, with the flight pathway, we're going GF, PSI, DLMN to DLM muscle. So those are the two pathways that we'll be looking at to see how that affects the escape response latency. So now we were talking about interneurons, we were talking about motor neurons, but if you don't have a neuroscience background, um, here's a little bit of review for that. So motor neurons relates to muscle. So these are neurons that are going to be connected to muscle. Um, and some basic neuron terminology is you're going to have your cell body, and then you're going to have your dendrites here going down to your axon, and then your synaptic endings. And the reason why I bring this up is because some of the parameters that they're going to be changing relates to dendrites and these different anatomical features, so it's important that you do know them. So that's the motor neuron. And now here we have the interneuron. So same type of structure, except that we're not connected to the muscle this time. Here's some more things that were mentioned. So we talked about gap junctions. So this relates to the electrical synapse. And here, down here, you can see these blue things here. This is your gap junction. So this is what's going to be connecting these two neurons, um, versus the chemical synapse. We see that it's different. And here's a bigger overview of the connection between two neurons. All right, so now we're going to brainstorm a little bit. So we talked about some things that can possibly cause this increased escape response latency. Um, but now I'm going to turn it over to y'all and we're going to split up and we're going to try and brainstorm what um, what may cause this. So either different parts of the anatomy, maybe it's something chemical, maybe maybe it's even the fly's mood. Like I just want you to brainstorm and to think. So let me get some breakout rooms set up. We'll do four of them. And when you're in your breakout room, go ahead and scan this Google Doc and put your ideas down there. Try and come up with at least five ideas. So we'll be gone for about, let's say, 5 minutes. And I'll be popping throughout the rooms to check. And then we'll come back and see what we got. So okay, all the rooms should be open. Welcome back, everyone. Looks like we had some good discussion. Seemed like everyone was participating, which is great. We'll go ahead and um, if we could have one person from each group share their ideas. So if we could have room number one go.
Okay, I'll go ahead and read what we wrote down. First, we put age-related breakdown of the giant fiber, then difficulty in cleanup of the brain as aging goes on, and proper conduction as aging continues, decreased plasticity in the neural circuits, degradation, degrade of the reuptake and other synaptic processes. Aging may allocate resources to other behaviors outside of the escape response.
Perfect. Thank you. Those are all some great ideas. Breakout room number two up next.
For break room number two, we said the shape of neurons affecting the speed as they age, reduce muscle or neuromuscular movement, Syn- synaptic transmission slows down over time, slower neural signaling as neuron membrane ages, diminishing number of interneural gap junctions, and developing a form of Alzheimer's or memory loss with age.
Perfect. More great ideas. All right, breakout room number three.
So, we talked about chemical signals throughout over time. The example that we use was like a light bulb, how the lead wires, they wear out, and so they lose that, signal for the electrons to pass through. So that's the kind that we use. And for two, we mentioned loss of neurons in the face could lead to late response. As aging goes, maybe they lose neurons. The longer the neuron body cell, the longer it takes to deliver the message. I mean, that's one thing too. The longer it is, especially if they don't have myelin sheath, which that speeds up the conduction of the conduction. Yeah, which is what we talked in number four too. And then the fifth is that some flies might develop mutations in their genetics. So it could be that, I don't know, they might not have as many neurons as a normal fly would have, or it could be anything.
Yes, more good ideas. Thank you. All right, breakout room number four, finish this off.
So we thought that there's a decrease in neurotransmitters with an increase in age, so it decreases the ability of the sensory neurons to transfer signals to the motor neuron. We thought that also harmful chemicals might cause like an allosteric shift in the motor neuron or like the sensory neuron, which inhibits the ability of the signals to be transferred between them. We felt like they could also develop mutations over time, so the receptors in the channels change with age, and natural degradation of the neurons with age.
Perfect. Thank you. All right, great ideas, everyone. So now this is kind of like the same process that these researchers were going through when they picked out different parameters that they wanted to test. So we'll see which ones they chose and then what results that they got. So I'll go ahead and share my screen again. Okay, so now what parameters did they choose? How did they do it? We'll go into that next. So modeling neurons. So when we're modeling these neurons, all of this model is based off of real physiological properties from these fruit flies. So we can see over here on the left, um, all of the different papers that they used to find these properties, to find the numbers for their properties. And then using all of these different numbers, they then put them in to this mathematical model to get these latency response times and to see how they're different in young versus older um, fruit flies. So you'll also see that some of them were estimated because at the end of the day, the model that they're using is supposed to predict real data. So some of them have to be estimated um, in a specific manner to make sure that the model is describing accurate features from these fruit flies. And there's a specific way to do that in computational neuroscience as well. So we see that this is all based in real data. And there's different Python packages that help people do this. So the paper used Neuron in this case, and we'll be using Brian too to replicate it in a different manner to see if we get the same results that the paper did. All right, so now going into interpreting the results. So this is the second figure in the paper. Again, we see this diagram that we saw earlier, and we see these different, um, this diagram of the difference between young and old fruit flies. So we see that this latency is longer, so their reaction time is slower than the young ones, which makes sense, right? These are gonna escape faster than the old ones. So with this figure is just to say, hey, the model is working how we think it's gonna work. And down here, what they're also looking at is the gap junction conductance. So I know in some of the groups we were having people mention conductance. And what this is talking about is how easily this current is able to flow through that gap junction. So if we have a gap junction, you may think like, oh, if we have a gap junction that's bigger, um, that's going to allow more current to flow through, and then maybe the response latency will be quicker, or maybe like, oh no, maybe if it's smaller, then the current will flow through faster, um, and then it'll be quicker. So these are the different questions that they were setting out to answer using this model. And we can see down here that based off of the gap junction conductance, as this conductance increases, we see that the latency is decreasing, meaning that they're responding faster. So this is showing, okay, gap junction conductance is an important one to be looking at. And we see as well that when you look at the gap junction conductance of a young fruit fly versus an old fruit fly, the young fruit flies have a higher gap junction conductance versus old ones. So this is what this figure is talking about here. And the reason why we're focusing so much on gap junction conductance is now in the future figures, they're going to compare gap junction conductance to other physiological and anatomical properties to see which one is more important in finding the escape response latency. So we'll look at those figures now. Now, I'm not gonna lie, these figures are kind of confusing at first, but don't get discouraged. They're called codependency analysis. So like we talked about earlier, we're looking at two different features and seeing which one describes how we can find the escape response latency better. Which one is more important, you could say. So in these um, codependency plots, the gap junction conductance is going to be on the y-axis for all of them. And then we have different features down here. So this is for like sodium, potassium, and then the leak. And we also see in these plots that we have a tiny blue dot in each of them. And this is going to be for our young fruit flies. So we see that they have higher conductances than this orange dot, which is our old fruit flies. So again, this is from the data that we were looking at earlier. So higher conductance, they're going to respond faster. Makes sense. Now, what these lines mean are, can we move along one of these lines to get up to these young fruit flies? And into this red dashed boundary zone which is kind of like our good zone that we're trying to get into so that we can say, okay, is there any parameter that we could change that could restore this quick latency from old fruit flies so that they could have this young response latency. So now that's kind of like a lot to wrap your head around. Or, me too. Um, so in this first plot, in all three of these plots, spoiler, the answer is no. So there's really no line here that this orange dot is sitting on that we could easily get up into there. Um, and what that's telling us is that in this case, gap junction conductance is more important um, than if we were to change these other parameters. And specifically, this is looking at the TTM. So now they did the same thing, except looking at the DLM, so that other branch. And again, we're looking at this line and seeing, okay, is there any way we can get up here? No, not really. Okay, so we're kind of going over here and it's starting to go up, but still nowhere near that. Again, same story here. So again, we're seeing the same story that um, changing any of these is not having as large of an effect as if we were to change the gap junction conductance. So then they asked themselves, okay, well, if we were looking at the physiology, the sodium and the potassium, what if we looked at anatomical features? So some of the things that they looked at were the diameter of the motor neuron or uh, the lateral dendrite or the medial dendrite or the axon. So they're looking at different anatomical features now. And what we're starting to see is that we see these kind of more curved slopes, right? Um, and what this is telling us is that, okay, some of these features are having a more equal effect of these gap junction conductances. So this one you see this diameter, we can almost get up here, um, but still it's requiring a lot of change of this diameter to get into that red zone. Um, and out of all of these different anatomical features that they were changing, they found that one had the biggest difference, and that was with changing the PSI diameter. So if we remember PSI, that was the peripheral one that we saw earlier. And we can see with this one, if we travel along this line, we almost get into this red dash boundary area. And none of these other plots, if you do that same thing, does that occur. Some of these other ones you get close, but this one you get really close. So what does that mean? That means that if you were to change the PSI diameter of these old fruit flies, you could restore their escape response latency back to their younger selves. So it's kind of like you're almost reversing their aging in a sense. It's one piece of the very complex problem, but this is the big moment for the paper is that we're saying, okay, there's something that we can change to restore this latency. So what does this mean in real life? Well, now, if you're an experimenter, you could say, okay, how could I change this diameter? How could I shrink this diameter? You could say, well, what if I alter a gene that shrinks this size? And then now we have another experiment that we can do in the future to see, okay, if we really do shrink this gene, does this restore um, and essentially hopefully reverse this aging? Who's to say? But with computational neuroscience, um, we're able to figure out these problems and test these things that couldn't be tested before. Like you wouldn't be able to do this if you in real models of fruit flies unless you were spending a crazy amount of money. Because imagine changing all these parameters in actual fruit flies. And to do so with such precision, going from like 5 to 5.4 to 5, it would be a crazy amount. So this is the powerful tool of computational neuroscience. Now, this last figure, um, before we were looking at oh wait, hang on. I apologize. This is the figure that we're just looking at. Let me look at the paper. Everyone can still see my screen, right? All right. Um, so now before, this is the last figure. You remember that we had the gap junction conductance on the y-axis each time. Now we're looking at what happens if we keep the gap junction conductance constant and these other features, we change them. And with that, they were also able to identify some other features um, of importance as well. And we can go more in depth with those features later into the program. So that's an overview of the paper. Um, again, just some big concept things. We're looking at what things can we change to restore this escape response latency from, these old flies to back to their younger selves. Um, and we're able to do so using computational neuroscience. So that is it for the paper. If you have any questions, please let me know. Feel free to ask them, put them in the chat, send me a message later if you want to. And then for upcoming events, we have we are planning on doing a workshop every week. So next week will be an open science workshop. So this will be more general, just talking about open science principles and reproducibility versus replicating a paper and the ethics of open science. Why is it beneficial? What are some barriers to doing open science? Everything like that. Um, and we'll be having more of these workshops while everyone continues to build up their Python coding skills and their knowledge with GitHub and everything. Um, and that will be mostly independent work. We went back and forth on whether or not we wanted to do in a more group setting or a more individual setting. But honestly, the best way to learn how to code is just by doing it. So I sent out that Scrimba course. It should be free, and it does an amazing job of taking you learning how to code and to do it all so that you're familiar once we start using the neuron packages. And also to do the GitHub course because GitHub is what allows everyone to be able to work together with their code and to collaborate. So those are two essential skills to learn. Um, and then once you do finish those courses, send them to me. And then I've been tracking everyone's attendance with everything for those student stipend awards at the end of the semester. And I'll be sending out kind of like a leaderboard, just so everyone knows where they're at with the points. Um, and then also please remember to do your baseline test so that we can get everyone assigned into groups. And this is for attendance today. Thank you all again for attending live. Thank you for attending live. Um, I'll leave that up there for a little bit. And once you get that filled out, you are free to go. Thank you so much for coming. And if you have any questions, let me know. Otherwise, have a great night.
Appreciate it. Thank you.
Thank you. Have a good one.
Bye, thank you.
Bye. Thank you.
Thank you, Ethan. Good night.
Thank you. Have a good one.
Thank you. Good night.
Bye. Thank you. Have a good one.